So it's a great pleasure to introduce our next speaker this afternoon, Ania Jayich from UC Santa Barbara, and she will tell us about a very innovative imaging technology here with diamonds.
So thank you very much to the organizers for inviting me here. This is really a pleasure to be here. And in the spirit of this symposium, the Diamond NV Center is definitely a novel sensor. In fact, some of the first biosensing applications with NV centers have only been realized within the last 3 years. And in fact, even though we think it might be potentially useful for neural imaging, there haven't been any demonstrations of imaging neurons yet with the NV center. The diamond community, in particular, and nitrogen vacancy community, is really trying to figure out what is the best possible way to image biological systems, and also what sort of systems would be the most amenable to this type of nanoscale sensor. And that's why it's really nice to have these types of interactions. Particularly, I've been very happy to have some seed funding here from the Center for Brain Activity Mapping, and working together with our Mark Ellsman and Ken Kosik up at UCSB. So this has been really fun for me. That's why I love my job, because you're always learning every day. So, okay, so Confucius once said that a diamond with a flaw is better than a pebble without one. So that might be true, but I think that a diamond with a flaw is better than even a diamond without one. In particular, if that flaw is the nitrogen vacancy center in diamond. It is the workhorse of our lab because it has uniquely accessible quantum properties, really excellent quantum properties that are accessible in a simple setup, at room temperature, in air, with a standard confocal microscope. And furthermore, the NV Center is very sensitive to a wide variety of fields, such as magnetic fields, electric fields, temperature, strain. And because of its very small size, it's in fact atomic scale size, it can sense these fields with very good spatial resolution. And so today I'll tell you about some experiments from the community and some few initial experiments in my lab using the NV Center and applying it to biosystems. Okay, so the big picture goal is not too different from that of other neural sensing schemes where one would like to transduce a firing action potential into, say, an optically detectable signal. So we envision decorating neurons with very small, say 5 nanometer size diamond nanoparticles containing fluorescent sensors. And as a transmembrane potential propagates along the neuron, that produces a longitudinal current. And as you might remember from physics, longitudinal currents produce magnetic fields that circulate the direction of the current. And the idea is that the NV Center would be used to sense the magnetic fields associated with these propagating currents. The currents are of the order of a nanoamp that would give magnetic fields on the order of tens of nanotesla, with which it should be possible to detect these with the NV Center. As I said, based on the sensitivities of NV centers that we've demonstrated in our lab as well as other labs. Of course, this hasn't been demonstrated yet, but this is the ultimate goal. And to do this quickly with sub-millisecond response times, which would be necessary to get a good spatial temporal map. And the idea, there's sort of 2 types of modes of operation we imagine. One would be sort of simultaneous monitoring of signal propagation in a whole neural network, as we saw earlier today, where one could do some wide field imaging and look at a large area of NV centers all at once. Or something that I think is somewhat interesting, although it doesn't seem to be as, I guess, sexy in the community, is monitoring individual ion channels, partially because that's really where the NV Center shines. Because of its nanoscale spatial resolution, you could really actually envision looking at individual ion channels opening and closing. Okay, so at the outset, let me give you some insight as to why we'd consider diamond as a potential material. Well, I've just told you that it plays host to this nitrogen vacancy center, this really nice quantum sensor that is small, it's fast, it's very bright and photostable, similar in some ways to what was shown earlier with the upconverting nanoparticles. Diamond itself as a material is biocompatible, and it's non-toxic to cells. There's a rich surface chemistry, and there's a large, actually, I've sort of been learning more about it in preparing for this talk, a large area of research devoted to functionalizing diamond surfaces and targeting specific biological cells, parts of a cell. And lastly, small, on the order of 5 nanometer sized nanodiamonds can be made in a controllable way. And they can contain NV centers that are exist in nanodiamonds down to the 5 nanometer size. Something I'm not going to talk about today, but just as a side note, there are other types of defects in diamonds that people are starting to look into too. One in particular is the silicon vacancy center, which actually people have seen fluorescent individual silicon vacancy defects inside of 1 nanometer size diamond nanoparticles. That's a topic for a different discussion. Okay, so though neural imaging has not been experimentally demonstrated, there have been a few beautiful experiments recently that have really shown the power of the NV center as applied to biological, or at least sort of what I call wet and squishy systems. I'm trained as a hard condensed matter physics physicist. There's sort of a divide in condensed matter physics between hard and soft, and so in this particular experiment, there was 2 experiments actually that came out around the same time, in which these 2 groups detected an NMR signal from a very small volume, about 5 nanometer cube volume of nuclear spins housed in a living inside of just a thin film of oil. Okay, I'll actually show you some experiments from our lab later that give you a little bit more detail on this type of experiment. But the point is that this is doing magnetic resonance imaging, or NMR, on a volume that's much smaller than anything that's commercially available in standard magnetic resonance imaging today, which would be more on the tens of, state of the art would give you tens of microns of spatial resolution. And the ultimate goal here is really to get to the single nuclear spin limit, and sort of the goals of, partially goals in my lab, as well as, say, this is our collaborator Dan Rugar at IBM, are to actually image the structure of proteins by doing nuclear magnetic resonance on the individual nuclei. So this is a really nice step in that direction. A little bit earlier, there was a demonstration of injecting fluorescent nanodiamonds containing NV centers into worms, C. elegans. In this case, these worms were fed some nanodiamond-containing food, or they were injected and imaged for several days, and the worms did just fine, and the NVs and the fluorescence was maintained over many days. So this is just an indication of the sort of non-toxicity and the photostability of NV centers. These little red guys are the NV centers. And lastly, and maybe most recently, there was a recent nice work demonstrating NV-based detection of magnetotactic bacteria. So these little bacteria that actually have magnets inside of them. And they use these for navigation. And by putting these magnetotactic bacteria on top of a bulk chip, 2 millimeter by, for us, okay, bulk is 2 millimeter by 2 millimeter chip of diamond, they were able to probe the NV center and probe the local magnetic field to image the magnetic field coming out of these magnetotactic bacteria. So here's just an optical image, and here's a magnetic image where one can clearly see the correlation between the cells and the or the bacteria and the magnetic field. Okay, so quick outline. I'll tell you a little bit about the background and the physics of the NV center. I'll tell you about about how the NV center actually senses. What's the mechanism for sensing? I'll go through a few of the challenges for biosensing, of which there's several, and I'll list a few. And then I'll tell you briefly about some experimental progress in our lab towards eventually bioimaging with NV centers. Okay, so what is the NV center, the nitrogen vacancy center? Diamond is a carbon lattice. An NV center is a defect in the carbon lattice, where one carbon is removed and replaced by nitrogen, and right next door there's a carbon atom removed, and that's not replaced by anything, it's a vacancy. And these can occur naturally, and they do occur naturally. In fact, defects in diamond and other stones are really what give give diamond its colors. So NV center would make diamond sort of pinkish, reddish. So they do occur naturally, but one can also cause them to occur by implanting nitrogen and subsequently annealing the diamond. By annealing, the vacancies become mobile, and they kind of like to land near the nitrogen. They form a nice stable atomic-like complex there. They can also be formed through growth. Actually, that's something that we specialize in at UCSB. I'll tell you a bit about that later as well. Okay, so this little defect complex, as I mentioned, forms an artificial atomic system. And like some atoms, it can emit photons and absorb photons. Okay, so it has an optical transition that allows us to detect the presence of the NV center. And also, like some atoms, it has an associated electronic spin. Okay, so maybe you remember from your physics classes what a spin is, but if one just thinks of it classically, it's like a little magnet. Okay, and so the spin is what I'm talking about. The spin is this ground state energy. Here I'm plotting the energy levels of the NV center. These two bottom levels are the ground state. The splitting between these is on the order of megahertz, and those two levels constitute my canonical two-level system, a little magnet that can point up or down, which would be like being in this state or being in this state. And this is important because a spin, which is like a magnet, responds to magnetic fields. And so, for instance, if you put a magnet in a magnetic field, it will precess, and it will do so at a rate that tells you about something about the strength of the magnetic field. And that's what ultimately gives us our sensing abilities. And I already mentioned that this atomic-like system exhibits quantum properties, meaning that it retains quantum behavior in ambient conditions up to on the timescale of milliseconds, which is really quite remarkable for a solid-state quantum system. Okay, so I can briefly tell you about how these are experimentally addressed. As I mentioned, it's a pretty simple setup. We just build our own confocal microscope where we illuminate the NV center through a high numerical aperture microscope objective. We illuminate the diamond sample with green light. The NVs in turn fluoresce. They emit photons in the red. And that is collected by some very sensitive photodetectors. So here on the left is a confocal image of NV centers where we scan the laser across a few micron size area of the diamond. And these bright spots each correspond to a single NV center. So the point is that single NV centers are, and we can confirm this through techniques and actually that were referred to earlier, anti-bunching techniques from quantum optics, where we can really confirm the presence of single NV centers. The point is that these are bright and individual ones are easy to locate. To address NV centers, we optically address them, but we also perform optically detected magnetic resonance, which necessitates a means by which we can deliver microwaves, about 3 gigahertz microwaves to our sample, which we do by patterning the diamond with waveguides or the glass which the diamond sits on with waveguides. And then at that point, we fix our laser on one spot, and then we drive a set of microwave pulses that we basically have drawn from the NMR community back in the 50s in order to exert quantum control over the NV center and do sensing in that way. Let me just show you quickly what it looks like. So it's not your typical confocal microscope that you might buy from Nikon, partially as physicists, but also because there's a few customized things that we want. It basically is a microscope with its guts splayed out all over optical tables. There's just a bunch of lenses and mirrors. These are microwave circuit here. Here's a single photon counting detection where we have 2 APDs. And then this is just a zoom in of our little diamond sample, which sits on top of a glass cover slip with a waveguide patterned on it. And we have some RF connectors coming in to deliver the microwaves to the sample, just to give you a flavor of what the type of experiments look like. Okay, so to try to describe a little bit how the sensing works, I mentioned that the ground state energy manifold of the NV center is a spin. It's a 2-level system. And we can spectroscopically address the splitting of this two-level system through optically detected magnetic resonance, and relying on the fact that when the NV center is in the lower state, it's much brighter than when it is in the upper state, and that's necessary. I won't explain why that is, but that allows us to spectroscopically probe this splitting. So in the absence of any magnetic field, we have a 3 gigahertz splitting between these two levels. We do optically detected magnetic resonance, and we see this signature at 2.87 gigahertz. In the presence of a magnetic field will affect this energy level here, which is actually doubly degenerate, and split it into two. And the amount of the splitting is directly proportional to the applied magnetic field. And we can see this peak split into two, and the amount of splitting is exactly proportional to the field, and hence we can infer what the magnetic field is. An important thing to know is the sensitivity, our ability to measure very small changes in the magnetic field, is going to depend on the line width of these peaks. Okay? And the line width of these peaks is fundamentally limited by quantum decoherence, which is sometimes called T2 star in the NMR community. And that's one of the reasons why the NV center is a good sensor, is because it has very narrow lines. Okay, so we've used the magnetic sensitivity of the NV center to do nanoscale magnetic resonance. So what we've done is we've taken a bulk diamond sample with NV centers a few nanometers below the surface, which we can controllably place them there. And if an NV center is only a few nanometers below the surface, its detection volume is on the order of the depth cubed. So what we did is we, in this case, looked at an NV center that was 10 nanometers below the surface. We put a drop of immersion oil on the surface. And those and the immersion oil has hydrogen spins in it, and those proton spins, in the presence of a magnetic field, as I already alluded to before, are going to feel a torque, and they're going to start processing around the magnetic field, just like a spinning toy top. And they'll spin at a rate that's proportional to, well, this particular gyromagnetic ratio of the hydrogen in this case, as well as the strength of the magnetic field. And that characteristic frequency produces an oscillating magnetic field at the location of the NV center. And that characteristic frequency can be resolved in a type of measurement. Here, this is for the experts in magnetic resonance, this is a XY8 pulse with 128 pulses that we've applied to the NV center. But basically, we're probing the coherence of the NV center as a function of wait time. And then when the Larmor precession frequency of the proton spins is matched to our wait time, we see a signature, we see a feature in our NV coherent signal. And so this is a coherent signal that is due to this 10 nanometer cubed volume of nuclear spins in the oil in this case. So yes, this is immersion oil, but for us this was sort of, we're getting closer to these wet and squishy systems. The nice thing here is that oftentimes one quotes and one talks about the sensitivity of the NV center, but you're talking about an NV center that's inside of a bulk piece of diamond, which is very well protected from its environment. But here we've really brought our sensor right near the surface, and actually in the presence of fluctuating spins and whatever's on the surface. And so this was a nice demonstration that it works. And the total number of spins just that we're detecting in this 10 nanometer cube volume is 60,000. Again, we're trying to get to the single nuclear spin limit. And we think, actually, that there are pretty good prospects of getting there within the next year, we think. OK, so just to summarize the different types of magnetic field sensing that I've talked about, there's this NV-based detection. In this case, it was just detecting static magnetic fields. These magnetotactic bacteria are little ferromagnets, and they produce a magnetic field, and the NV centers can be used to detect those. Us, as well as other groups before us, have done NV-based NMR detection of a nanoscale volume of external spins. And then the goal eventually is, and there was a proposal 2 years ago out to do this, to detect the magnetic field associated with currents flowing down axons. I just want to point out that these experiments have mostly been done in bulk diamond. I mentioned the prospect of nanodiamonds, and that's certainly an exciting prospect. We need to work towards that. Currently, the properties of NV centers inside of nanodiamonds are not quite as good as those inside bulk diamonds just because there is so much surface. The surfaces are notoriously messy, and they have significant effects on the coherence of the NV centers. It's not just surface, though, because we, in this experiment, showed that we can make NV centers as little as 3 nanometers below the surface that still have good coherence properties. And I'll actually talk about the issue of using nanodiamonds and making better nanodiamonds shortly. But so far, most of the detection sensing measurements have been done with bulk diamond. There is this one exception, and also another demonstration of another type of sensing. Is a recent paper by the Lucan group at Harvard, where they used NV centers for very sensitive thermal sensing. And it had been shown previously in the groups of Dima Budker at Berkeley and David Avshalom, who was at UCSB, now at Chicago, that the NV center can detect temperature changes of 1 millikelvin per root hertz, meaning a 1 millikelvin or 1 millidegree change in a 1 second averaging time. And this was used in this case where I think these were some embryonic fibroblasts, human embryonic fibroblast cells, where they injected nanodiamonds into the cell. A, B, C, D, and E are different nanodiamonds. Then they put a little gold nanoparticle inside of the cell, and they heated up that gold nanoparticle, and then they measured the temperature at locations A, B, C, and D just by probing the NV centers at those locations. And they see that the temperature dies off with distance from the heat source, as one would expect in the right distance-dependent fashion. So this is sort of a really neat nanoscale thermometer as well. So I think this is exciting, an exciting direction to go as well. Okay, so what are some challenges? I already alluded to this fact that if one wants to have very high spatial resolution and good sensitivity, you have to bring your sensor very close to whatever you're trying to detect, which means you have to form very highly coherent, quantum coherent sensors near the diamond surface. So here's, if one were to take a bulk diamond, if let's say you have some target spins that you're trying to do magnetic resonance imaging on, or if it's a current flowing down a neuron, you still want your sensor to be quite close to that source of magnetic field. Your sensitivity, or in this case, if there was a nanodiamond with something around it, or likewise, your sensitivity to magnetic fields is given by this equation. The important things to note here are it goes as the distance cubed and the coherence time. The quantum coherence time. So really one wants to have small r's and large long T2s. But these traditionally don't go hand in hand. The closer you bring your NV center to the surface, the worse it gets. And so this is a challenge that a lot of the people, in particular in my group, are really focusing on. One way in which my group is focusing on is through the creation of the material. So there are several ways to get diamond. One could go to Cartier and buy, spend a lot of money on such a diamond, or you could spend even more money actually and buy one of these. It's called a CVD, a chemical vapor deposition system, and this was a diamond growth system that was purchased by David Avshalom and really set up by his students when he was at UCSB. As I already mentioned, he's since left. But the idea here is that you can grow very clean diamond with very good control. It's a bit slow, 8 nanometers per hour, so we only grow about 100 nanometers of diamond, but we grow that on top of a clean substrate of diamond, but we really only care about that last 100 nanometers. Some other advantages of the system is that we can actually introduce nitrogen during the growth. So we grow the diamond, and then we introduce the nitrogen in a very thin layer where we can really control the depth below the surface down to nanometers. And in this way, we're introducing the nitrogen without crystal damage. So the way that almost everybody does it is they implant nitrogens with a high-energy ion implanter. That's very damaging because in the process of ions bombarding the diamond, they create lots of vacancies and damage. This is a much gentler technique, and it also really allows us to position these in depth with nanoscale positioning, and it gives long spin coherence times. Here's some data that we've, together with David, have published on the coherence times of NV centers that are right near the surface. So at a depth of 5 nanometers, we have coherence times of about 200 microseconds at room temperature, which is quite long. even for NVs. Of course, we do see that, as expected, the T2, the coherence time in this case for 50 nanometers is a factor of about 4 larger. We do see a consistent drop in T2 in the coherence and the quality of the NV center as we bring our sample near the surface. This is something we're actively trying to understand. It's actually a very interesting problem, what's going on at the surface. And in fact, using the NV center is a very sensitive probe of whatever junk there is on the surface. To that note, I should mention that, sort of taking a step back, but when I talked about the NMR detection of oil on the surface, if we remove the oil, we still see a small little signal. It's weaker, but that's actually just detecting a layer of, we think it just could be a thin film, nanometer thin film of water on the surface. So the sort of sensitivity of the NV is almost a double-edged sword. We're sensing everything that we even don't want to be sensing. Another challenge, as I mentioned, is if one wants to really do in vivo imaging, you want to be able to make small nanodiamonds and inject them into cells. And so there have been demonstrations of 5 nanometer nanodiamonds with fluorescent defects inside. These are traditionally produced in a rather violent way. It's detonation nanodiamond. They're basically an afterproduct of some explosions. But they can be made quite small, and they can be sort of sorted to get reproducibly small nanoparticles. This is a TEM image showing a bunch of 5 nanometer-ish size particles. But as I mentioned, the sensing properties are severely degraded. Again, the question is why. It's an interesting question, and it's a not known. Another approach that we're taking in the lab is actually starting with a really clean piece of diamond that we've grown and doing top-down fabrication, so actually etching nanoparticles. In this case, we're etching nanopillars. So this is a SEM, a scanning electron microscope image of sort of an army of little nanopillars of about 200 nanometer diameter, and they're a few microns tall. One can make these smaller. This is our first demonstration. And we've shown that NV properties in these are superior to those in nanodiamonds. Another nice benefit of this is these pillars actually act to waveguide the fluorescence out of the pillar, and therefore increases our collection efficiency significantly. Another challenge is functionalizing nanodiamonds to bind to specific sites, say inside a cell. This is basically all I'm going to say about it. There are people working on this, and as I mentioned, it seems to be a pretty well figured out system. You can attach proteins and polymers and make the surfaces hydrophilic, etc. Okay, so in the last 10, 15 minutes, I'll tell you about some recent experiments in our lab with the goal of, I guess, bioimaging. So we've heard a little bit today about gadolinium, the gadolinium ion, and its use as a contrast agent in magnetic resonance imaging. So here's, for instance, a MRI slice of the brain. On the left is just a standard MRI image. On the right is an image using gadolinium as a contrast agent. When that happens, this tumor here lights up. The gadolinium was allowed to cross the blood-brain barrier here. The way this works is the gadolinium is a pretty large spin. It's a big magnetic moment. It's a 7/2 magnetic moment. But moreover, it's a spin that's rapidly fluctuating. So it's basically flopping all around with a characteristic timescale in the order of nanoseconds. So it's flopping around very quickly. And because it's doing so, if you bring it nearby, say, water molecules, which is what NMR is generally detecting, it causes those water molecules to also start relaxing. So basically, this quickly fluctuating gadolinium spin causes any nearby water molecule to relax. Okay, and so this is sort of called relaxation imaging. And this is what we will use as well for the NV imaging. But I guess that shows up next. Let me also tell you one other thing about gadolinium is its use as a spin label. So there are techniques called site-directed spin labeling where one can specifically bind gadolinium complexes to certain parts of, say, a protein. Okay. And in this case, there's an experiment where they were able to detect the distances down to sort of the nanometer scale between gadolinium spin labels. to image something about the structure of the protein. In the experiments that I'll talk about, we'll primarily make use of this relaxing properties of the gadolinium. But instead of the instead of the gadolinium relaxing proton spins as it does in these images, we're going to have the gadolinium be relaxing the NV center spin, and we'll use the relaxation of the NV center as our contrast agent. However, this is also interesting because this plays a role in my very futuristic view of what we're going to do, which is to take a protein, attach it to an AFM tip. That's fairly easy to do. A big part of my lab is involved in scanning probe microscopy, so this is one reason why we are envisioning this. To take a protein and specifically label it with gadolinium spin labels at different points, scan it over an NV center, and then when the gadolinium comes near the NV center, the NV center feels it's getting relaxed, and we can in that way image the structure of the protein. Okay, so that's my futuristic goal of this experiment. I already mentioned that the way that gadolinium works is when it comes near to, say, a proton spin, in our case the NV, it causes the NV to relax more rapidly. And so the way we do the experiment is here's sort of the time scale, the chronology of what happens in our experiment. We turn a green laser on. I didn't talk about this, but by shining a green laser onto the NV, we can initialize, we have, again, this two-level spin system. We can initialize our spin into one of the two levels. And these two levels, remember, have a different level of fluorescence. So we initialize our system into one spin level. It becomes bright, and then we turn off our laser. And then in the absence of any light being shined on the NV center, it will slowly relax to its equilibrium state, which is some mixed state between these two levels. And it does that with a characteristic time scale, which is called the relaxation time. Okay. In the presence of gadolinium, though, if we were to, say, zoom in on this part of the experiment down here, so without any gadolinium, we might get a characteristic relaxation curve that looks like this. With gadolinium, that will enhance the relaxation of the NV center, causing it to relax more quickly, and get the blue curve. And so if we fix our measurement time to, say, in this case, 2 milliseconds, and look at the fluorescence of the NV center after letting the NV evolve during this dark time, we should see a change in the fluorescence signal. Okay. So that's what we're looking for. So we're looking for a change in the fluorescence of the NV center when a gadolinium is brought nearby. I should mention that there was an experiment last year done in the group of Jörg Wachtrup in Germany where they used gadolinium, this type of relaxation imaging, to image just the structure of a cell. They took a HeLa cell and labeled it with this gadolinium complex, which also had a fluorescent marker attached to it, this Alexa fluorescent marker. And so here, if they just take an optical image of the marker fluorescence, they see the outline of the cell because this complex is specifically attaching to the cell membrane. And then they took a T1 image so this cell, by the way, is sitting on top of a piece of, again, it's a piece of bulk diamond in this case. Again, nanodiamonds have not quite there yet for the sensitivity. So it's sitting on top of a piece of bulk diamond. This image is basically probing the NV centers and looking at the relaxation time. And one sees that you get sort of a dark feature where the relaxation of the NV center is increased in the same region where you see the fluorescence, confirming that one can do T1-based imaging. So as I said, we want to get to the point where we can do scan-probed imaging and scan some protein or other structure over an NV center. And so we wanted to study systematically the NV relaxation dynamics as a function of this NV to gadolinium distance. So we took an AFM tip, a silicon AFM tip, functionalized it with gadolinium, and scanned it over with 30 millimolar gadolinium, scanned it over an NV center with about a 1 nanometer amplitude, tapping amplitude. So here is, this is a combined confocal AFM microscope that we've built. So here is a confocal image showing the outline of our AFM tip, and all of these spots are single NV centers. And so we can control will be position our gadolinium-soaked AFM tip to within nanometers of the NV center. So some of the data shown here, when our tip is far away, say a few microns away, we measure the T1 of the NV center, and we see that it's about 5 milliseconds. Then when we position the tip directly over the NV center in much closer proximity, and this NV center, by the way, is about 6 nanometers deep below the diamond, so the gadolinium and the NV are separated on the order of 6 to 10 nanometers. We see that the T1 is reduced to 4 microseconds, so we get a 3 orders of magnitude change in the relaxation time of the NV center. So then to show this a little bit more clearly, we took an image. So this is an image where we're scanning 60 nanometers in one direction and 1 micron this way. And in the color scales plotted the T1 of the NV center as this gadolinium tip is scanned over the NV. And one sees that the T1, where it's red, is again almost 3 orders of magnitude smaller than when the tip is far away. So we can really start imaging the gadolinium. We can take a line trace across this data and plot T1 as a function of tip position. And we see that we have a pretty broad feature, but our spatial resolution can be estimated to be about 50 nanometers, which is the characteristic size scale over which the T1 changes by about 3 orders of magnitude across that 50 nanometers. So again, the goal is to image a single gadolinium spin. So we've started in this direction by working with Craig Hocker's group at UCSB, who's a chemist. What they've helped us do, or actually what they've really done for us, is to take a silicon AFM tip, functionalize it with a PEG polymer chain. These PEG polymer chains can be, you can buy them in different sizes. We bought some in the 6 to 10 nanometer size, and functionalized this AFM tip with PEG polymer chains, and each polymer chain has a single gadolinium attached to it. So in the end, what we're expecting is that we have a cantilever coated with gadolinium, but they're on the order of 6 to 10 nanometers apart, and we're hoping to be able to resolve those individually as we scan them over an NV center. That's what we're going to do. And just to confirm that this functionalization worked, we have really nice facilities at UCSB, multi-user facilities. We used a SIMS, a secondary ion mass spectroscopy measurement, where we shoot ions we took our AFM tip that we think has gadolinium on it. We shot ions at it, looked at the emitted ions, did mass spectrometry on those, and confirmed the presence of gadolinium. These are all peaks associated with the gadolinium, with the expected gadolinium atomic mass. And we also see that the signal saturates after about 8 nanometers of drilling material away, which is consistent with there being about a 6 to 10 nanometer PEG layer. So those are the next set of experiments that we're going to do. And just to give you some idea of what types of sensitivity we expect, if we can bring that single gadolinium to within 4 nanometers of the NV center, which we can make NV centers within 2 to 3 nanometers of, we can grow them within 2 to 3 nanometers of the surface, we should be able to get a SNR, a signal-to-noise ratio of 3 in about a 3-second measurement. For a it's very sensitive to separation. For 6 nanometer separation, it would take us about a minute to get an SNR of 3 to detect a single gadolinium spin label. And other prospects, including NMR detection on the spins that are in this polymer, in this PEG polymer chain too. That's another thing that we're thinking about doing. Okay, and so the last bit of data is very preliminary, and maybe it's embarrassing to show because it sort of shows how we are not really bio-experts. But we've started to try to integrate our confocal microscope with neuron imaging. And again, this is sort of our future. This is our artist view of what would happen. We would have a diamond sample. Each of these are little spins inside with neurons plated on top, either stretched across or grown on top. And the idea would be to use wide field imaging to look at many NV centers at once, image those onto a CCD camera, and look at sort of a spatial temporal map of what's happening in the network. So together with Ken, and again, they really did the growth for us, Mei Zhang and Ken Kosick's lab at UCSB, grew rat hippocampal neurons on diamond, so that took a little bit of playing with the parameters. But this is an image of a neuron grown on top of diamond. We fixed these neurons, or they fixed the neurons. I didn't know what fixed neurons were before, but now I do. And so these are some confocal images of these neurons taken in our setup. Okay, so really this just shows that the neuron growth is successful. But the one thing that is maybe a little bit concerning is the autofluorescence is quite high. The Cossack lab was sort of surprised to see this, but I think we have very sensitive detectors, and so we have to work on reducing the autofluorescence of the neurons. However, one potential reason this could be is because, from what I understand, that maybe fixed neurons fluoresce more brightly. So to not have to use fixed neurons, and eventually we'll want to do sort of live imaging, we've built this little live cell imaging apparatus. Again, this for us was not the usual type of thing we have in our lab, but here you can just see our glass coplanar waveguide, our RF coming in to drive electron spin resonance of the NV center, our little perfusion cell. It's an inverted optical microscope. You can see the little green dot right there where the laser comes in from the bottom and focuses on the NV center. This is integrated onto a heater stage to keep the neurons happy. And there's, yeah, a standard perfusion cell. So our first experiment was we just quickly grew some, or again, Mei, Zhang, and Ken Kossack's lab grew some 293T kidney cells because they grow very quickly, just to see if we can keep cells alive. This is an optical image of our cells on top of our coplanar waveguide. You can see these little bumps corresponding to what we think our cells are about the right size. We took an optical image of the cells. There seems to be some features here that are cell-like. There is, again, some bright fluorescence from these cells. Ultimately, we maybe don't care about the 293 T cells, but we don't know why these are so fluorescent, if that's because they're coming off, or I don't know, maybe some people have some suggestions. But if we do take, and these are cells grown on the diamond, if we look at an area where there's very little autofluorescence, potentially no cells there, we take a confocal image, and this is a confocal image showing NV centers, which is almost identical to the image that we took before plating with neurons. So the nice thing is that the NV fluorescence is unchanged from before the cells were grown on the diamond. The other nice thing is that the 293 T cells, although they're bright, they appear to stay alive during imaging. We confirmed this by putting some stains in afterwards to make sure they were alive. If this autofluorescence is going to continue to be a problem, there's ways around this. We could utilize lifetime imaging, utilizing the fact that the NV center has a relatively long lifetime compared to the general fluorescence lifetime of most likely of what the neurons have. And we could also use turf microscopy, and that's something that we are working on together with Mark Elsman here at UCSD. Okay, so in summary, I hope that I've shown you that NV centers and diamond are promising biosensors. There's been some nice demonstrations and some exciting proposals. They're small, biocompatible, fast, photostable, and sensitive. I've talked to you about a different type of imaging. There's lots of different types of modalities of imaging that NV centers can be used in. I've shown you some relaxation imaging, which has promise for detecting spin labels. And even though the challenges are plentiful, the field is young, and so we think that there's some excitement up ahead. This is a picture of my group. Really, the people that were involved in this experiment were an undergrad, Anand, a new postdoc, Matt, and Brian, a graduate student. And I also want to thank my collaborators, Mark and Ken, for teaching me everything that I know about neurons. So thank you very much.
Thank you, Anna. That's exciting. A wonderful new technology with spatial and temporal resolution to die for. Terrific. We're open for some questions. Let me perhaps begin by asking about the manufacture of the NV diamonds. The bulk diamond seems to be the preferential way to go. You can generate those, I guess, fairly reliably. Can you generate the very small nanodiamonds that one might want for an in vivo application? Yeah, can you generate NV centers inside of them? Yeah. Yes, you can. You can also do irradiation. You can do implantation with nitrogen
And one can also just start with nitrogen-rich diamond and do electron irradiation to generate vacancies. So that's, and one can generate NV, like nanodiamonds with, say, 50 nanometer diameter that have hundreds and hundreds of NV centers inside of them. Again, kind of similar to what the nanocrystals that Jim was talking about.
So well, let's wait for the microphone. If we, yeah. Oh, very good. Yeah, Jim, go ahead.
Yeah, thanks. Beautiful stuff. I guess my question was somewhat related to what you just said, actually. So when you have a lot of NV centers that are very close to one another, do they talk? Do they cause each other to decohere? Can you win by having multiple ones?
Yeah, so you win up to a point, and at some point, the dipolar coupling between the nitrogen and the NV centers are going to limit your coherence time. Basically, you can get flip-flop processes, and yeah, so you can't go too dense. So you win as, like, your sensitivity goes as the square root of n, the number of NV centers that you have. Actually, another kind of big problem is that the conversion ratio of nitrogen to nitrogen vacancy centers is not 100%. So if you keep adding more nitrogen, you just, yeah, you stop winning after a point.
So Ania, first a criticism. How do you control so that the only students that join your lab are as tall as you?
You know, I was actually looking at that this morning, wondering what happened, because I think I'm the tallest. I think I must be standing in a hole or something.
Yeah, it's staged. So the real comment was, and you probably know this literature, I only know because I visited, there's a group in Melbourne that is doing the same sort of construction, and they showed me a project, I'm sure it's published, using the pillars as an appliance to solve
but more like as an electrical.
It's a retinal, yeah, it's a retinal recording
Yes.
system,
Yeah.
which they're promoting as a way to actually replace a missing connection to the brain. So they're actually doing implants, making a big deal out of it with those kinds of pillars. But those pillars look like, if we could address them more individually, which of course we can, as a pretty good substrate for at least looking at slice preps or whatever.
Yeah.
So you might want to say something about what you know about the Melbourne folks, I guess.
Yeah, I mean, I heard them give a talk in China. I know there's a lot of questions from the audience about why are they actually doing it with diamond, what's better about the diamond than the silicon, for instance. I don't know, I mean, so again, that's something I can't really speak to because I don't, but apparently silicon wears quite well in the body too. And so, yeah, I mean, it's certainly, I mean, so diamond is biocompatible. Obviously, it's more fun to make things out of diamond than, or to sell things out of diamond than silicon. And so a few things that diamond can be also made electrically conductive. I haven't talked about that at all. But by doping diamond with boron, in fact, you can even make diamond superconducting by doping it with boron. But diamond can be made conducting, and that could be used for individual connections. And so that's certainly a potential. That could, even be mixed together with this type of magnetic imaging. And especially if one can grow, I mean, so we can grow the diamond, we can introduce impurities during growth to dope it. It's hard to dope it N-type, but to dope it P-type is not so hard to do.
Yes, it's quite beautiful, but it's essentially a magnetic field detection. And no matter what its properties, if you were detecting magnetic field, you are sensitive to all the neurons in the vicinity to which they contribute magnetically.
Yes.
And so it's not too easy to interpret on a packed neuronal system. As opposed to a single neuron, you can interpret the magnetic field. But in a collection of populations of neurons, you seem to me to have a very difficult time decoding the signal. Right. Because the magnetic fields just add according to the known laws of physics. And so it's sort of like doing MEG. And when you have any collection of neurons, you have great difficulty in disentangling their magnetic field contributions from each neuron. And
But I think one significant
Isn't that
difference
right?
from MEG, I think, no, that's absolutely correct, but I think one significant difference is that your NV center is right next to one of the neurons, and the fields die off as 1 over, for a field going across down, current propagating down a wire, the fields die off as 1 over R. So being 100 nanometers away from one neuron, you're probably going to be
a lot stronger for that neuron.
You're going to be a lot stronger for that neuron than from a neuron that's, I don't know what your next neuron is going to be, maybe 20, 30 microns away. And that's going to have
20 to 30. Well, I
don't know. You tell me.
Well, unfortunately, neurons are often close packed, and so they're.
But it's close. Yeah. So how? But are they like within hundreds of nanometers of each other?
Well, they're practically touching. So
that
separated by a thin layer of glia, but their initial segments will be big signals.
Okay.
Yeah. So okay. So that's certainly a good point, and that would be that would not help.
Yeah.
I haven't talked about detecting the electrical. NV centers could also. I didn't, for the interest in brevity, didn't talk about this, but NV centers can also detect the electric fields that are generated by these membrane potentials. It's not as sensitive of an electric field detector, and one loses a little bit by the the Debye screening. If your NV center is outside of your neuron, you have your electric fields die off quickly. Magnetic fields don't die off as quickly as the electric fields because they don't have this extra Debye screening. But I think in the beginning, when I sort of said we're still trying to figure out what is the best way to detect neurons and what is the best system to do it in, these are the exact type of situations. For us right now, our first experiment is sort of putting neurons in a not really real setting, just plating them onto a chip in which you wouldn't have these issues. But that's, I guess I didn't realize that they were that close together in the brain, so that's good to know.
Also, a completely different question. Is the relaxation of a single spin really exponential, or is it all or nothing? Isn't the graph you showed of the exponential die-off of T1.
It's all or nothing.
All or nothing.
It's all or nothing, but our signals are averaged.
Sure.
It's a temporal average of many.
Temporal or spatial ensemble
It could
averaging.
be either one, actually, right? It could be either one. So when we look at a single NV center, it's a temporal average over many instances.
Yeah, okay, but a single one at genuinely single spin is either all or nothing. That's
no, there's, yes. Yeah.
So the question is there any polarization sensitivity in the Zeeman effect that you have in the NV center? Any polarization sensitivity?
Oh, of the magnetic field. So it's only sensitive to the component of the magnetic field that lies along the NV axis.
Okay. So do you have, when you do magnetic field sensing on living cells, for instance, do you control as well the orientation of your NV centers, or?
No. So in bulk diamond, you can control it to some extent, well, to a very good extent, because there's only 4 possible orientations, and actually one can grow the diamond to grow them along one orientation. For in the cell, no, you don't have control. Your nanodiamond is going to land wherever it does, but at that point, it's fixed. And so, unless you get really unlucky, there's going to be some component of the field, and it's, on average, you're only going to be down by, say, a factor of √2 for the, in terms of down from the best possible scenario, in which case the magnetic field was aligned along the NV axis. If it's, you might get really unlucky and it's aligned exactly perpendicular to it, but then you just wouldn't use that NV center. Especially if you have a lot of NV centers to choose from, and the idea would be to have a ton of NV centers, and you're looking at some average of them as well.
Good. Well, let's thank Ania again for very interesting